Good evening. We're starting our first session for Unit 4. If you are on Zoom, just anytime you have a question, raise your hand or I'll be sending you questions. So make sure that you answer these questions, okay? So, what this lesson is about? It's about the first topic of Unit 4. But before we get there, I would just like to show you an outline of the entire unit. So, you could plan according. So, first thing we're going to be doing today is photosynthesis. We're going to split photosynthesis between today and next week. So today is going to be the first part called light dependent reaction. So the same photosynthesis you took in IGCSE, we're going to take it a bit more details. So that's why we're going to have one part called light dependent and one part called light independent reaction. That's for next week. Okay? So let's call this topic 1. So that is topic 1 of Unit 4. Topic 2 is going to be ecology. Ecology is the study of environments. And we're going to split topic 2 into first, energy flow, meaning we're going to learn about food chains, how does energy flow in a food chain. Second is called sampling. Sampling is like a practical thing you do to study an environment. Third is succession. Probably never heard of it. It's some series of events that lead to change in an environment. Next comes global warming. You're familiar with this. And next comes evolution. So look, we're not going to do these in separate lessons. No, this and that is one lesson. These two in another. Okay, but nonetheless, they all fall under the same topic of ecology. So how many topics we've done so far? Photosynthesis and ecology. Next comes this topic called forensic biology. One of the nicest topics you'll study in this lesson. Forensic biology is how to use your knowledge of biology to study crime scenes. And another topic underneath is called DNA profiling. You see, I've put this under forensic because you can nowadays use something called DNA fingerprint. Meaning use your DNA to identify people, to identify criminals, to identify victims. Next comes, also quite interesting, immunity. Those who go into medicine, they will find this very, very useful in the future. With immunity, we're going to split into two as well. One is called non-specific. This is your natural defense systems, meaning your skin, your mucus, tears, everything you have in your body to protect you against disease. Specific is the ones where you use antibodies, use white blood cells. Okay, so we're going to do these in separate lessons. Finally comes microbiology. Microbiology is a branch of biology where you study little things. Mainly, we're going to be studying here bacteria and viruses. Okay? So this is what Unit 4 is all about. We're starting today with the first topic, as I said, light-dependent reaction. Okay? Everything is fine so far? So, next step is I'm going to show you what part of your specification I am going to be doing. I'm going to do the overall reaction of photosynthesis. This is something you have done in the past, like the equation of photosynthesis. Structure of chloroplast, you've done this too in Unit 2. These are new stuff. So we're going to learn what colors, what wavelengths of light plants prefer to grow. Those of you who've done chemistry, probably familiar with chromatography. I'm not gonna confuse you with these, but these will soon become clear. So I'm gonna show you how do plants make ATP and how do they break down ATP. So let's get started with just like an overview of photosynthesis. The best thing to start from is to look at a plant in action doing photosynthesis. Here you go. What I see here is some plant been exposed to the light. I want you to take a close look to what's this plant doing as it's exposed to the light. If you zoom in, you'll notice, whoop, look, bubbles. Do you guys know what gas bubbles these are? These gas bubbles are bubbles of product of photosynthesis, that is oxygen. Okay? And usually you can actually determine the rate of photosynthesis by just counting how many of these bubbles are produced per unit of time. Okay? So if you see too many bubbles, that's higher rate of photosynthesis. Less bubbles, lower rate of photosynthesis. Okay? And I sent you a PDF this morning so we can write along with one another. I want to write the word equation of photosynthesis. So what are the raw materials? There are two raw materials of photosynthesis. One is carbon dioxide. Two, it's water. Now, there are two necessary conditions for this to happen. Number one is a source of energy, light energy. Next, you require a catalyst. The catalyst here is chlorophyll. Products. You got glucose. You got that, we call it the waste material of photosynthesis, but it's not very accurate. Oxygen is also used by plants for their own aerobic respiration. Okay? Now I'm going to go into more depth showing you how the raw materials get into the plant and what would happen to the products of photosynthesis. So let's get into more depth. So first is water. Every time water flows in biology, do you guys still remember what we call the process by which water flows from one area to another? That is always gonna be osmosis. So it's through the roots by osmosis. Then comes CO2. CO2 is a gas, so you'd expect it to be diffusion. Where it's going to enter the plant, this will always enter the plant at the stomata, in the leaves. Now, I've color-coded the elements here. So see that the hydrogen is in blue, the oxygen is in purple, carbon is in red, because I want to track the same atoms in the product. So, glucose. Take a look. Where does that carbon of glucose come from? Obviously, it's going to come in from carbon dioxide. What's really important here is that where does the oxygen in glucose come from? The oxygen in glucose comes from the carbon dioxide. So the source of oxygen in the glucose, which is the valuable product of photosynthesis, is actually carbon dioxide. You may tell me, like, look, there is an oxygen here in water. This oxygen is gonna be diffusing out of the chloroplast. Okay? Next comes oxygen. As I said, this oxygen at the end comes from the water. So once again, you have water, carbon dioxide, glucose and oxygen, which will escape from the chloroplast. It might be used for aerobic respiration, or it might be just diffusing out of the plant. So in brief, the process will look something like this. Plant, light energy, combining water and carbon dioxide to produce glucose and oxygen. Now let's check what is going to happen to the glucose. Let's see what are possible destinations for glucose from photosynthesis. So, number one, you could simply respire the glucose. We forget this quite often, that plants have to respire. They need to get their energy requirements, and that's done from glucose they make. Number two, it could be stored some later on, or it might be even, you still remember this word? Translocation. So they could be translocated as not glucose. You guys still remember what we call the sugar to be translocated? Not glucose, definitely not starch. Do you know what it is? It is sucrose. So if it's that sugar to be transported around, it's going to be called sucrose. Okay? So based on that, we got three sugars. Glucose, that's product of photosynthesis or reactant of respiration. Sucrose, that's transported, like we call that translocation. Finally, for later on, you may store as starch. Okay? All good so far? Now, in that specification, they wrote something called phosphorylation and hydrolysis. Now, you took two reactions in Unit 1. These are the only reactions actually we talk about in biology. One is called condensation, one is called hydrolysis. It's just like this condensation reaction I'm going to tell you about right now involves phosphate. So for that reason, we're going to call this condensation reaction, we're going to call it, listen, phosphorylation. Say it, phosphorylation. So we're going to phosphorylate a molecule. So let me show you how. 1, 2, and 3. This is, do you recognize this? This is exactly what you have in DNA. That's a nitrogen base, like adenine in my case, attached to some pentose sugar, 5-carbon sugar, and from here I'm having a phosphate, another one, okay? Guess what I'm making here? This is what we call ATP. Whoops, it's actually written. So ATP is that molecule. I want to show you how do we use it and how do we build it. What I've done here is called phosphorylation. When I attach a phosphate to it, that's called phosphorylation. Phosphorylation is a reaction that gets input of energy, meaning it's endothermic, meaning it would require energy to be supplied. Okay? When you store in energy, that's called phosphorylation. Okay? However, you may reverse this reaction, meaning you can take that phosphate at the end and break it down. When I break down ATP, I actually form ADP. Okay? Meaning I am releasing energy. One more time. ATP as it is like this, that's a charged form. It's storing energy. Most of that energy is stored in the last bond between phosphate number 2 and phosphate number 3. Okay? At the moment is highly rich with energy. But in this form, ATP is lacking energy because I lost that last bond here where the phosphate was attached. And that moment I can recharge. So look, I can always go from this form to this form This way I call it phosphorylation. This way I call it hydrolysis. You do that same thing inside your body. You continuously recycle ATP. So time when you are in the morning, you charge up, you have a meal, you make a lot of ATP. At night, when you're exhausted, you're lacking ATP and you are rich with ADP. Now, phosphorylation, as I said, is as simple as attaching a phosphate to ADP and turn it to ATP. Today's lesson is about photosynthesis, right? Meaning I'll be using the photo part of photosynthesis, which is light. That's why the phosphorylation we're going to be doing today is going to be called, read this along with me, this is called phosphorylation. Okay? So I think I misspelled it. Photosynthesis, like this. What does photophosphorylation? Photo, you know that part. Photo is light. Phosphorylation, you attach a phosphate. So photophosphorylation is one word, meaning you use light energy to phosphorylate, to attach a phosphate to ADP, turning it into ATP. One more time. Here you go. This is what I call phosphorylation. General, it occurs in humans, animals, plants. But when it involves light, like the one I have here, I simply add the word photo. Okay? So for animals, we require food to phosphorylate. In plants, they just do it with the help of light. Opposite is known as hydrolysis. Okay? Is it all good so far? Okay. I need now to give you some chemistry background. So assuming that you haven't done chemistry, or just a reminder of some chemistry basic we need for today's lesson. Mine here is called redox, oxidation reduction. You need to understand this in a very basic form. Okay, so if you're not good at chemistry or you forgot all what chemistry is about, you really don't need that. What I want you to understand is just two things. Number one, you see this guy here? I wrote under it, has been reduced. To reduce something in biology, we assume that it's gaining hydrogen. So if something gains a hydrogen, then it's getting reduced. Opposite is opposite. If somebody loses this hydrogen, then it's getting oxidized. One more time. In the context of biology, oxidation reduction is quite very simple. It will only involve gain of hydrogen is reduction, loss of hydrogen is oxidation. You may tell me, like, why hydrogen? What does hydrogen have to do with this? Simply because hydrogen is the simplest element in the universe. When I say hydrogen, I mean a proton and an electron. Meaning, if something gain of electrons is reduction. And the electron here is inside that hydrogen atom. Okay? So like in chemistry, we usually say oil rig. Oxidation is loss of an electron. Reduction is gain of electron. But since we're not into those tiny little things in biology, we just assume it's hydrogen. Okay? So that's one thing I want you to understand in chemistry. Another one, I want you to realize that there is no oxidation without reduction. They must occur at the same time. So redox is called redox for a reason, because they must occur simultaneously. So You may tell me, like, how can this always happen? How can oxidation reduction always happen? That's luckily inside our cells, we have something called coenzymes. What does a coenzyme mean? A coenzyme is co means like co-worker, company, something to help enzymes. So in cells, we always have some carrier of hydrogen whose job is to snatch a hydrogen from molecule and attach it to another. So basically, this guy here is the one who's involved in redox. We call it again, we call it a coenzyme. The one we're interested in today is called NADP. So this guy basically takes that hydrogen from one molecule and attach it to another. Meanwhile, it's getting molecules oxidized and reduced. You may tell me, like, what the is NADP? It's not necessary, just called NADP. Okay? NADP, coenzyme, it's to carry hydrogen from one molecule to another, right? So that's our carrier. You need to know that's the one, because the hydrogen is not simply going to fly in the air. No, we need something to carry it around. Okay? All good? Now, to get further, before we get into more depth on how photosynthesis is going to happen, I just want to remind you of the leaf structure. And you've probably done that. You must have done that, actually, the structure of a leaf. So we're going to take a side view on the leaf. So that's a top view. So I want to show you how a leaf is from this side. To start with, every leaf must have a layer looking like this at the top that we call the waxy cuticle. This is what's meant to protect the leaf, prevent water loss. Right underneath, I have what I call epidermis, like a skin, transparent, to allow light to penetrate. Okay? Next comes the layer that is in charge of photosynthesis. Most photosynthesis takes place right here, called the palisade mesophyll. That's like the queen. Every other layer is serving this layer, trying to maximize photosynthesis. Right underneath, I have a layer that is containing air spaces. We call it the spongy mesophyll. This layer has few chloroplasts, but is not the main job. The main job of spongy is to let the CO2, the gas, move all the way to the queen, the palisade. Okay? Here at the bottom, I got two openings, stomata, and that's how the CO2 comes in and moves out. Why is this turning off? Let's try to turn on. Turns on. Okay? So, once again, the leaf structure, we got the waxy cuticle to prevent water loss, epidermis transparent, letting the light pass through, spongy, that's the layer that allows gas exchange. I got the palisade, that's where most photosynthesis happens. I still need the vessels, so you got two vessels. One at the top called xylem. Xylem is the one transports water to stir sucrose translocation. Do you remember that part? So that's occurring here at the bottom of the vessel. Okay? Let's try this. Who could help me out trying to complete this part? Anyone? Raise your hand. Here you go.
Hi.
Go ahead. Hi.
I cannot see the screen.
Why not?
Oh yeah, that's
Okay.
no, I cannot see anymore.
What happened?
I don't know, it's black.
Okay. Can I try from my side?
Yeah, that's it. No, it's gone again.
Okay, second. I think when you speak, it disappears. So, just a moment. Let's try this here. I messed it up completely.
That's fine.
Is it, do you see it better now?
Yeah.
Okay. Or still flink?
Stomata allows gases in and out.
Mm-hmm. Yep.
Chlorophyll absorbs light.
So, yes. Chlorophyll absorbs light and
Broad transport.
you're gonna put this one here, right? Veins. Large area. That is.
Thin.
Large area means broad, right? So the leaf is broad, so it can absorb more light. Thin, short, diffusion, distance.
Thin.
Can I show you again? So you got openings like your nostrils, they allow gases to move in and out. We know that chlorophyll is a pigment to absorb light. Leaves being wide is such a good thing because it absorbs more light energy. Veins got the xylem and phloem in it. Plus, veins are supported because xylem is tough tissue to keep the leaf upright. Finally, thin leaves are thin. That's a good reason because the gas can diffuse much faster when the diffusion distance is short. Okay. Looking now at the chloroplast. So, chloroplast is an organelle that is highly adapted for photosynthesis, meaning every little thing in this organelle is designed to maximize the rate of photosynthesis. Now, there's something you have to realize to understand how this organelle works. You have to realize its origin. We so much believe that chloroplasts and mitochondria at some point in history of life were actually bacteria, prokaryotes, meaning this is like a primitive cell. It engulfed at some time in the past a bacterium. That bacterium found benefit staying inside the cell because the cell protects it. And it starts generating ATP for both itself and the cell. And this turned to become mitochondria. Another bacterium was engulfed by our larger cells. This bacterium had chlorophyll. The bacterium benefited because it's protected, but meanwhile, because it's photosynthetic, because it got chlorophyll, it got engulfed and they stayed together to the moment. What evidence do we have about this origin, the bacterial origin of chloroplast or mitochondria? Look, bacteria has one membrane, we know that, right? When it got engulfed, the vesicle where it got engulfed got its double membrane. So that's one thing. Another very good, someone's telling me the ribosomes within the mitochondria or within the chloroplasts are small, reminding you of their bacterial origin. One more time. Chloroplasts, special thing. It comes with small ribosomes. See those spheres there, the little balls in gray? This is the ribosomes. In addition, I have circular DNA. That's again typical for bacteria, and double membrane. The double membrane tells you that there was a vesicle and a bacterial membrane. So all of this, how many evidence do we have? We have three. The double membrane, small 70S ribosomes. Third is the fact that we have circular DNA. Yes, so chloroplasts. Inside, you got some stacks. You see all these dish-like stacks? They look like this, like imagine them like this. These are plates where you have the chlorophyll. So each one of those plates is what we call thylakoid. What's a thylakoid? Individual plate. The entire stack of plate, the whole stack, is what we call granum. Okay? So one more time. The chloroplast has individual membrane, we call them thylakoid, and they're usually stacked one on top of another. Each stack is called granum. And those thylakoids, they work just like solar panels, meaning they're not fixed in one orientation. They may tilt, they move about, they try to get as much light as possible. So do you see these other membranes here? Like this, like this. Those are known as lamella. What are these membranes? These are membranes just to position the thylakoids in their best orientation to grab as much light as possible. Okay, so how many new words you've learned? Thylakoid, the membrane. Lamella, the attachment that puts the membrane in the right position. And a stack of thylakoids is called granum. You may see it somewhere as grana. What's the difference? Grana, many. Granum, one. Okay? Plus, all of this is floating within a jelly-like substance. Remember, cytoplasm bacteria. Here we're not going to call it cytoplasm. We're simply calling this whole stuffing inside the chloroplast, we call this stroma. So again, you got stroma, the jelly stuff inside of it. We got lamella. We got thylakoid. And a stack of it is called granum. Grana in plural. There is always double membrane. There are small ribosomes. And there is always some circular DNA inside, reminding you of its origin. It would be wise if you draw it. So if someone asks you to draw it, how are you going to draw it? Draw double membrane, inner, outer membrane. Together you may call them an envelope. Small ribosomes, thylakoids like this. Stack of thylakoids is a granum. And this whole thing is like stuffed with stroma. Okay? Any question? Here you go. Let's try this. Let's try this here. I want you to match either one of these with the correct letter. Okay? So let's do this in the chat box. I show you the feature and you guys tell me which letter it is. Okay? Number one is stroma. So where am I gonna put the stroma? Which letter is showing you stroma? It is actually letter C, right? Which one is pointing at the thylakoid? So which label is pointing at thylakoid? That's a right. Go. It's D. Next says most make photosynthetic enzymes. This is not direct, but I want you to think about it. Think about it. Enzymes are like what? Enzymes are like proteins. Agreed? So in other words, where are proteins made? Easy, right? Okay, that's A. Absorbs light energy. Which part of it absorbs light energy? That's where chlorophyll is found. Remember the plates that got the chlorophyll there? So I must be pointing at the thylakoids. Small ribosomes. Codes for enzymes. Code. Doesn't make no code for the enzyme. So what am I supposed to say here? Codes for them. That means it is the DNA. Next says carbon fixation. What does that mean? This is something I want you now from now to understand that within the chloroplast itself, we are gonna divide it. So the chloroplast itself must be divided into two parts. One is where we're gonna do the light reactions of today. Light dependent reactions, meaning those are the reactions that require light. That's gonna happen all in the thylakoids. Okay? And the other one is stroma. Stroma is where we're going to do what we call light independent reactions, meaning reactions that can occur in the light or the dark. Independent. So that's what we call in biology, that's like a general term, doesn't only apply to chloroplasts, it applies to everything. It's called compartmentalization. It's a long word. Compartment is section, meaning the chloroplast is subdivided into two sections, two rooms, compartments. Each does separate function. One is to absorb the light, and one is to fix carbon dioxide, is to make the sugar. As I said, this is next week. We're mostly interested in what we call the light reaction. But for the time being, I just want you to understand that carbon fixation, meaning making of glucose, takes place in stroma, while light reactions take place where the chlorophyll is in the thylakoids. Okay? Once again, someone says light independent reaction. Where is this supposed to take place? Once again, this exclusively happens in the stroma. Okay, so again, so far we've been looking at chloroplast. We found its bacterial origin. We said their evidence tells you that this used to be bacteria. Small ribosomes, circular DNA, double membrane. And today for the first time, we're splitting that chloroplast into one part here, the green one. We're going to call this the thylakoids. That's where the light-dependent reactions take place. Everywhere else, the stroma is where the carbon fixation, that's where CO2 turns to sugar. We call that light-independent reactions. Okay? Quick question. Can you guys answer this A, B, C, or D? Tell me. What do you think is the correct option? They are asking you about Z. So what do you think Z is? I can magnify it, zoom in to show you what Z is. You can see it on your phone. So again, we're interested in part Z. What is Z here? It must be what? It must be the granum, okay? Somebody has picked stroma. No, dear. This is one of the grana, okay? Stroma. Which letter would be stroma? They did not point a stroma here, unfortunately, but that's the granum. Now, we have a practical. This is the one where we're going to look at the pigments of photosynthesis and see how they do their job. Have you ever seen a plant leaf in autumn? You realize that chlorophyll, the green pigment, is not the only pigment that plants have. There are actually other pigments whose job is to assist chlorophyll absorbing light. Meaning, do you see here we have something orange, we have something yellow, and the chlorophyll. Usually we only see these in autumn because the plant gives up all of its chlorophyll, not much light in winter and autumn, and leave behind those what we call accessory pigments. So from here, I can see something called chlorophyll A, called chlorophyll B, and other orange, yellow pigments, we call them carotenoids. You may tell me, like, why do we have all these pigments? What's the point of having more than one pigment? I'll tell you, this is necessary because the more pigments, the greater number of wavelengths, the greater colors of light I can absorb with the plant. The plant will have more of the spectrum of light to absorb. So how does this happen? Let me show you how. Here is plant. So you see I have a light bulb on the left. I have a filter there in the middle. I'm going to change the wavelength. I'm going to change the color. And here I have some water plants and I'm trying to collect the oxygen. As I told you at the beginning, the very first thing in the class, I told you that's how we find the rate of photosynthesis. Meaning, the more oxygen, the more bubbles, the greater the rate of photosynthesis. So please take a look and tell me which colors of light are the favorites for this plant. So we tried purple, we tried blue, we're trying green. Whoops, not much is happening. When it's green, plants don't do photosynthesis, meaning they don't absorb it, meaning they reflect it. Once again, if the plant, look how many bubbles I got with purple, a lot, right? In blue, I got still a lot. When it's green, it's not doing any photosynthesis, meaning the plant is reflecting the green. And if you reflect a color, you will appear in that same color, meaning the reason why plants are green It's because they are reflecting the green light to our eyes. Yellow, still not a lot. Orange, somehow. Red, whoa, it's a lot. So let's take a look on the left-hand side and see the colors and rate of photosynthesis. Once again, keep an eye on the left-hand side now. See, a lot near the red region, less in the blue, nearly nothing in the green, then it increases again until we get to the red. So, in other words, plants prefer near the blue area, with the purple, and the red. They don't like it when it's green. They don't do much photosynthesis when it's green. And again, this has to do with the pigments that plants have. Look, this spectrum we were looking at is what we call an absorption spectrum. It's a spectrum that shows you what wavelengths of light plants prefer as they do photosynthesis. So you can see here that chlorophyll takes or absorbs one wavelength, chlorophyll B absorbs another one. Those orange pigments, they help also trying to absorb as much light as possible. And it shows you that this is where plants absorb light. Again, I don't want you to memorize these numbers. I just want you to understand them because they'll ask you about them in the form of like some scenario question asking you to analyze the graph. So if you've been asked to analyze, you're going to just say, well, this wavelength 400 to 500 is a lot of photosynthesis, and between maybe 600 to 700 is also high rate of photosynthesis. So this is what I call, again, I call this an absorption spectrum. You can link this to rate of photosynthesis, and that's what I call action spectrum. So again, what's the difference between this curve and this curve? Nothing, except what's written on the y-axis. Here it's absorption. Here shows me how much of the wavelengths are absorbed. Here, on the other hand, I have rate of photosynthesis tells you how much photosynthesis is taking place at each wavelength, and they perfectly correlate. You see? Exactly correlate. So where light is absorbed, that's where most photosynthesis is happening. Someone's asking, if certain colors are not reflected, like red, then why can we see leaves turn dark orange, almost red during autumn? They do turn orange and red near autumn, right? Because they reflect. Chlorophyll absorbs everything except green. The red-orange pigment, they absorb other wavelengths and they reflect to your eyes the red and orange. So once again, different pigments have colors which they don't absorb. Really important here. When a pigment is green, this means it's absorbing everything but green. When there's a pigment that is red or orange, meaning it absorbs everything except the red and orange. So your color is what you don't absorb. Your color is what you reflect. Let's try a question. They said, here is an absorption spectrum given to me. Underneath, they said, draw me an action spectrum. So what do? It's worth 2 marks. So very simple. Look, you take that same graph on top, and you repeat it to the bottom. You just have to follow the same pattern and you're done. Okay? You just need to show that they perfectly align and correlate. Okay? The height doesn't matter really. It's just need to match maybe with your ruler. You match these with one another. Yes, I have another experiment now, which is the chromatography. Here, we're trying to separate all the pigments that plants have. So let's do this. Let's say I gave you leaves. Usually in school, we do this with spinach because it's very, very rich with chlorophyll. So we take those leaves, we first grind them. We put sand or salt. Sand is better because it doesn't get involved. The sand is rough. So as you crush, it would make all these plant cells break. So you get like some nice paste once you add a solvent. The best solvent you can use here is ethanol or acetone. Okay? Now, to separate the pigment, to separate colors, we usually do chromatography. So I took some chromatography paper. I am going to put a drop of that paste onto the baseline. What's a baseline? It's just like a line I make with a pencil right here. Okay? Next, I drop the solvent like ethanol. I place that paper into that beaker, but very important, your paper shouldn't dip into the solvent. So let's say here is my beaker, here is my baseline. The solvent should always stay at the bottom, not touching the baseline. Got it? Like this. And here's my colored chlorophyll pigments put on the top. Soon you will notice that the pigments separate based on what? Based on the solubility. Meaning, the more soluble the pigment, the further up it will travel on that chromatography paper, something like this. So again, from here, I can see that this yellowish color here is the most solid. And here's the greenish one is less soluble. To identify those pigments, we usually do a small calculation called RF value. To determine the RF value of something, simply, you measure that distance traveled at the tip of the spot. Let's say mine here is like 3.8. And I divide this by what we call the solvent front. That's the distance traveled by the solvent. And in mine here is like 4.9. So 3.8 divided by 4.9 gives me a value of 0.778. That is what we call the RF value. And again, what's the importance of this? It simply, if you look up that pigment, you search it, you Google it, whatever you'd like, you can name it right after this. Okay, and I can compare it to some other pigment from another plant. So once again, how did we do this experiment? 4 steps: crush, dissolve, filter, then run in a chromatography paper. Okay, any question? All good? All right, let's now get into the main part of the lesson, which talks about like photosynthesis, the reactions of photosynthesis. Look, do you remember what I said about that the chloroplast is two parts? The one on the left here on your screen is what we call the light-dependent reaction. The one on the right is light-independent reaction. So again, here I have the granum and I have light been absorbed by chlorophyll. My reactant here is gonna be water, okay? The water enters this reaction and comes out from the other end as oxygen. So what am I missing here? I am missing the hydrogens. So again, water enters the reaction and gets through the chlorophyll, and oxygen is wasted while the hydrogen is what's been used. You may tell me, like, why is it that the hydrogen? Think of the product. What is my product? What am I interested in? I am interested in glucose. And glucose is simply carbon, hydrogen, and oxygen. The hydrogen here, that hydrogen comes, the only source of hydrogen I have is water. Okay? So basically, my target is to take a CO2 molecule and attach these hydrogens to it to make it a glucose. Okay? So again, my reaction here, my reaction is going to give me hydrogen. Okay? I need this guy here. Do you remember him? This was the coenzyme NADP. That's the one's going to carry my hydrogen from this end. From the thylakoid, is going to take it all the way to the stroma. Okay? Also, I'll be phosphorylating ATP because I need it. Soon you realize why it's needed. And those are going to go to the stroma. What's going to happen there in the stroma? As I said, we're going to take a molecule of carbon dioxide from the air, and this carbon dioxide will be attaching to the hydrogens with an input of energy to give me a molecule of glucose. Okay? So again, in brief, what did we do? What's this all about? This is an outline of photosynthesis. We're gonna split photosynthesis into two parts. One part takes place entirely here in the thylakoids, and another one takes place somewhere here in the stroma. Okay? The light reactions which takes place in the thylakoids involves H2O coming in, and it comes out on the other end as oxygen. It delivers hydrogen. It delivers ATP that comes from photophosphorylation. The light-independent reactions involving carbon dioxide, we reduce that carbon dioxide with the hydrogen, and it gives me glucose in return. Light reactions on the left, light independent on the right. I'll repeat this here. So, step one, light dependent reactions. So what's going to happen in step one? We are going to take a water. We're going to split that water like this. That's my water. We're going to split that water into its constituents, hydrogen and oxygen. Okay? The oxygen part, we know where it's going to go to. The hydrogen, remember I told you about hydrogen at the beginning of the lesson? I told you that a hydrogen atom is not one thing, it's two things. It is H ions, H plus, proton, and electrons, both of which are necessary, both of which will combine with carbon dioxide to give me glucose. Do you see what I wrote here? I wrote photo light, lysis. To lyse is to split. So this reaction of using light energy to split the water and get all the good things out of it is called photolysis, the splitting of water with light. Okay? So this is my light dependent reaction, like this. Get the water split, extract all the good stuff out of the water. I carry them with this coenzyme. We're now taking them to the stroma, where we attach that hydrogen onto the carbon dioxide and we make the glucose. Don't forget that you need ATP. Where would that ATP? This ATP is generated with light. We call that photophosphorylation. Okay? So let's try something. Let's try this question. They said, complete the diagram to show structures involved in light-dependent reaction. 2 marks. What am I supposed to draw here? Think about it. Again, they said, draw me whatever is involved in the light-dependent. So I really need to draw these guys, right? I am drawing thylakoids. So just gonna label thylakoid membrane. Okay, so I'm gonna show you what a thylakoid is. So that's the line here is the membrane, and this part here, which is not necessary for this question, we call the lumen, thylakoid lumen. And the entire thing, remember, the whole thing is called, good? So this is the 2 marks. You show granum, you show the thylakoids. Another question. Here I have a leaf, okay? And they said, why is it that's a good thing that the leaf is thin? Do you guys still remember what's the great advantage of leaves being thin? To allow rapid of CO2. So what am I supposed to say there? Good job. Which is needed for, from now on, every time you mention something, you mention a condition, or you mention a raw material, you tell them which part of photosynthesis it is. So, since it's CO2, do you guys say dependent or independent? Think about it. Because it says CO2, then I must say independent. Okay? When if it's light, I obviously have to say this time dependent. Okay? How about the midrib? What's a midrib? That's a midrib here, okay? Veins of the leaf. What do the veins contain? Midrib contains to transport water. What do we call that? Tissue to transport water. There is xylem, which provides not only water and minerals. Water provides me with what? Water provides me with electrons and protons. Which section? Was it dependent or independent? Think about it. Where did I put the water part? Let's take a look. Let's cheat. There, right? Midrib transports, because it's not only water and minerals, there's xylem, there's phloem. Do you guys still remember what sugar was the one that's carried in phloem? Sucrose. Okay, so let's now dig deep into their reactions. Let's do quick comparison between light reactions and light independent reactions, okay? Please, if you think it's a light dependent, put D in the chat. And if you think it's independent, put I, okay? Agreed? So let's start. Water split. Water photolysis. Where was this happening? Chuk. ATP is made. Where did I make the ATP? Was still there, right? ATP is broken down. Where is ATP consumed? We make it there, break it down here. Stroma was for which reaction? Was it for dependent or independent? By the way, that's the same thing. Carbon fixation, that's where we make sugar. How about the thylakoid? Where was the thylakoid used? Here. Sugar is made. Here or there. NADPH. Where did we mount the hydrogen onto the NADP? Where did we attach the hydrogen? Where we didn't put it's here. Okay? That's reduction. Where did we oxidize it? It's right here. We consume it right here. So let me clarify this one more time. Look at it. Here I made the ATP on the left. Light reactions, we made the ATP. Agreed? So the ATP comes here. We're going to remove that phosphate. We're going to use the energy. This is like a battery. It's going to charge up this light independent, right? So what will return back here? We will return back the ADP. Same. On the left-hand side, we reduced NADP. The guy is going to take the hydrogen here. But eventually, he will return back. So it's going to return back to the stroma minus its hydrogen. It's going to get oxidized right here. So it's going to come back here. And it's losing its hydrogen. Where did the hydrogen go? We use the hydrogen to reduce the carbon dioxide, making glucose. Okay? Yeah, so please pay attention to what's coming next. That's the final part. I want you to pay very attention to every little detail. They all really matter. So, where am I exactly? I'm in the chloroplasts. Specifically, I am standing right here. I'm standing on one of those thylakoids, okay? Outside is the stroma. Inside is that thylakoid membrane. Again, this is one of the thylakoids. That's the lumen we call. That is the membrane. And I'm outside. Okay? So It's called light reaction for a reason, because the light is gonna hit chlorophyll inside or on those membranes. So you see the light energy, and here is my, what I call, photosystem. What's a photosystem? Remember, it's chlorophyll, but we have other pigments with chlorophyll. So it's like a mixture of pigments. We call this mixture, call it photosystem. Okay? So what's going to happen at that photosystem? The light hits the pigments. When light hits something, it excites its electrons. That's why objects look shiny. That's why metals look shiny, because when the light hits the piece of metal, it reflects the light. It shows the electrons in the metal getting excited. Here's the same thing. Electrons in the chlorophyll get excited. What is exciting them? Exciting here is a scientific word. It's the light. It makes them go higher energy state, and they flow in like a wire, in like a circuit, from one protein to the next, okay? Why is this happening? Simply, this was rich with electrons, and the light excited these electrons. They moved in a circuit until they come down to their original state. So again, think of any object. Here's my object. I excite it, goes to a higher energy state. Now it has a lot of potential energy. We're not gonna drop it all the way. No, gradually we make it go down. As it goes down, it's actually giving me back that energy. If you hold in a pen right now, carry the pen up, drop it, you'll hear a sound. That tells you that you gave it energy. This is exactly what we did here. We excited the electron, it went to another higher energy state, and it did not drop all the way down. It's gradually moving down like this. While it's moving down, we are actually doing work. Instead of wasting that energy, we're gonna use that energy to do work. The work we're doing here is that we're gonna move hydrogen ions into that thylakoid space. How? Simply, I have enough energy. While this electron going down, it's giving me back that energy, and that energy is strong enough to pull hydrogen ions into the thylakoid, tiny space inside. Okay, again, let me show you how is this happening. Here you go. We're pumping hydrogen ions into the thylakoid space like this. How are we doing this? We're using the energy by the electrons moving through that thylakoid membrane. This is so much like this dam here. What do I have here? These are my hydrogen ions. I'm accumulating them like this behind the membrane of the thylakoid. Okay, accumulate them, build them up, and eventually I will let them flow out from a tiny hole. As they flow down, they will turn some sort of a turbine. It's literally a protein turbine like this you have in the membrane. And this, instead of generating electricity, it will generate ATP for me. One more time. How is this going to happen? Step one, what did we do? In step one, we excited electrons in one of those, we call them photosystems, okay? As we did this, the electron went from a higher energy state, excited electron went from a higher energy state to a lower one like this. Meanwhile, we accumulated the protons right here inside the tiny thylakoid space. Somewhere here on the left, on the right side of your screen, we got channel. We got a tiny hole. This tiny hole will work like the turbine. This channel will rotate. Literally, it rotates like this. And while it's rotating, it does phosphorylation. It makes me a lot of ATP. I'll repeat. So where am I exactly? I'm in the thylakoid membrane. What am I doing? I am simply exciting some electron, making that electron move through a chain. While it's moving, we're accumulating hydrogen ions like this. We build a gradient, we build a high concentration of this gradient. Soon after, those hydrogens are going to flow back through a turbine. And while it's flowing, it will turn that, generate some motion, kinetic energy. This kinetic energy will help me to make ATP. I know it's complicated, but that's why I want to repeat it. So, here we go. Step 1, light. Step 2, electrons. Electron flows from this excited state all through a chain. I'll be calling this chain simply electron transport chain. This is called, remember what we call this? We call this photo system. It's another photo system. There are two of them. This one here, we usually call it 2. This one here, we call it 1. Photosystem 1, photosystem 2. The electron moves from here till here, right? So while the electron is moving, remember what it does? It's not moving like journey. No, it does work. The work the electron is doing is to build up a gradient inside. I'll be calling this gradient electrochemical gradient. Why electro? Electrons. Chemical, because you got hydrogen ions inside. When these protons or H plus flow through that enzyme, the turbine thing, it will rotate it. And as a product, we will be forming ATP. Okay, so one more time, let's take a look. What I see here is photosystem 2. You always start with photosystem 2. It's light energy exciting electrons. The electron moves in the chain like this. Look, it's the electron now. It's right here. It's moving. While it's moving, it's pumping protons. You see? One more time. The electron moves through the electron transport chain and it pumps protons inside. That was PS2. That is PS1. What does PS1 do? Same as PS2. But they absorb different wavelengths of light. So now the electron comes here. We kick it up again. We excite it again. Some more light. All right. And then, do you see this guy here? That's the enzyme. That's the enzyme that makes ATP. We're going to call this enzyme ATP maker. Scientifically, we call it ATP synthesis to make. Because it's an enzyme, I call synthase. ATP synthase. So it's going to make me ATP. How? It literally rotates and it generates ATP. That's called phosphorylation. Okay, so one more time. Here you go. What do I have here? I got my photosystem 2. Remember, you always start with PS2 here. And electron, excited, moves through the electron transport chain, comes to PS1, gets excited again, right? And eventually this would help me to build what I call electrochemical gradient. The electrochemical gradient will help me to generate ATP. I'm missing just 2 bits or 2 pieces missing from this puzzle, which is, where do the hydrogen ions come from? To where will these hydrogen ions be removed? Because I need a supply to continuously run this stream. I need an input and I need an output. The input is going to be water. So again, what will provide me with the hydrogen ions is going to be water. See where water is? It's right here. Do you remember that photolysis part is going to happen at PS2? We split the water and we get the hydrogens. And I need something to sweep away those hydrogens, to remove those hydrogens. So here comes my NADP. The NADP gets reduced and it becomes NADPH. The best thing you could do now is to draw with me so we can summarize this whole process, okay? Draw a line like this. Stick some photosystems 1 and 2. So that's PS2. That is PS1, okay? I need some electron like this, and I need some carriers. 1, 2 carriers maybe here, okay? The electron is gonna flow like this, like that, through that electron transport chain. I'll give it some light here, photons of light, and light here. Labels: thylakoid membrane, okay? That's the thylakoid lumen. And what is outside? Stroma, okay? What am I building up here inside? I am building up a gradient of hydrogen ions. We call it electrochemical gradient. From here on the right, I'll make an opening. This is a channel. What do I call this channel? Called ATP maker or ATP synthase. Okay? This ATP synthase is gonna make photophosphorylation, meaning ATP. Here I have ADP and phosphate. Okay? I'm only missing one last thing, which is the inputs. I need hydrogen ions. So I'm going to put H2O split to 2H plus, 2 hydrogen ions. And to balance the equation, I'm just going to put half oxygen. Photolysis. Okay, so can we try to summarize it together? I'm going to give you the sequence of events, and you guys tell me what is the correct order. Shall we try? Can you please tell me what is the first event? Take a look at all these events here and tell me which one do you think is the first one? Tell me, what do you think is the first one? There's something before C. Someone wrote C. It's not C the first. There's something even before C. B first, right? After B is not C. No, wait, don't say C. Before C, there's something too. Because B alone can't happen unless something else is happening simultaneously. Think about it. What should happen along with B so it makes sense? That's right. G. Now comes your favorite. Yes. Okay. What comes after C? Good job, right? Perfect. Then goes to okay. Then is it A or E coming first? This says fine, all right? And this one goes right here. You may flip those two, doesn't matter, okay? Not make any difference. Okay. All right, we are done for the day. I will need you to maybe just stay. Please don't run away because I want you to join Google Classroom. So